Good afternoon. Dr. E.S. Krishnamurthy, my friends, invited guests here. It is a great pleasure for me to be here on this podium after a gap of few years. Myself and Dr. Krishnamurthy are associated with this program for many, many years, and we started that way back in 2000. And today, the TV Srinivasan's group has now jointly organizing this type of program at NIMHANS as well as in Chennai. So we are fortunate that NIMHANS is a part of the TVS Foundation's, the neurosciences group now. And that is why I am thankful for the TV Srinivasan and Venu Srinivasan and the family for giving us this opportunity. And I stand before you and bringing the greetings from my institute, the National Institute of Mental Health and Neurosciences, popularly known as NIMHANS. I am giving my own experience of nearly 3 decades. When I started in our own institute as a resident neurologist, I used to get many number of patients with a complaint which was similar to this. A 10-year-old boy developed staring expression, stiffened extremities, and loss of consciousness whenever he was given bathing. And this was the first description way back in 1945, came from New Zealand. But when I was working with my teacher, Professor who is very well known in the field as father of Indian epileptology, I used to see these cases one after another coming to me saying that my child is getting this episode. Whenever given bath, when the water is poured over the head, my child gets convulsions or fits, what we call as fits in a common terminology. The people were wondering, what is this? Is it a real entity? Some even went on to pull us down to say that this must be a Bangalore phenomena and nowhere it is seen. So the people started pulling also to say that this may end with Professor K.S. Mani. I have heard this year after year. But then I thought this is my opportunity to take it further and do the research and end all the controversies what is there. And now I am putting the whole thing as a nutshell in this form of presentation, starting from introduction to the reflex epilepsy, definition of hot water epilepsy, epidemiology of hot water epilepsy, clinical semiology, animal model, pathophysiology, pathology, EEG, imaging, autonomic functions, newer concepts in the management, and genetics of hot water epilepsy. The whole thing, we will look at that. Epilepsy is a very interesting and a challenging field for all neurologists. That is because there is a lot of stigma in the society. The society has made this disease much more curious for the common man. But what is this reflex epilepsy? The reflex epilepsy is a very interesting variety wherein you can produce these epilepsies with certain factors. That is called epilepsy, characterized by seizure with a specific modes of precipitation. That means you can produce this. How can you produce? Somebody having a switching of the lights can get a seizure. Somebody, when you listen to the music at a particular wavelength, you get a seizure. Somebody, when they are laughing, they can get a seizure. When they are crying, they can get a seizure. When they do mathematics, you can get a seizure. You may be wondering, what am I talking? Am I talking with some sense, or am I talking something which is a fiction? No, it is not a fiction. It is a fact that there are seizures which are precipitated by certain factors, and those are called reflex epilepsies. An understanding of this reflex epilepsy gives an idea for understanding of epilepsy as a whole. Because if I understand how the reflex epilepsy arises, I can easily understand why the epilepsy arises. And that is the understanding, and that is why people have used this reflex epilepsy model to understand more about epilepsy as a whole. What is this hot water epilepsy, which is a curious one? Epilepsy precipitated by the stimulus of pouring hot water on the head during the bath is known as hot water epilepsy. I'm sure all of you take hot water bath. I do not know in Chennai whether you practice more hot water bath, but definitely in Bangalore, all of us take hot water bath, which is a very, very common one. We also call as water immersion epilepsy or bathing epilepsy also. And this was known from many parts of the country, world. It is not that it is only in India. It was seen in Australia by Kippert in 69, Canada in 78, Japan by Kurata in 79, UK, Moran, USA, Stensman, and Turkey of recent years. In recent years, there are a lot of papers coming out from Turkey and Japan. And we are seeing that because all these papers are referred to me for evaluation. So I get to know even before publication that this paper is coming out. But what is interesting is it is frequently reported from South India, and that too from Bangalore and Karnataka, which was started with Professor K.S. Mani, my teacher and philosopher and a mentor. And then Professor Velu Murugendran, I'm sure you all know, he is a very well-known neurologist in Chennai. He has also published a paper in 1985 from Chennai. And that is the interest between Chennai and Karnataka. So we have done now the recent studies even to consolidate our findings. And this was a recent study which has been done from Bangalore, from nearby Karnataka. And we have seen that these are the places where we are more commonly seen. Southern part of Karnataka state, these patients are coming to, and we call that as Mandya belt. And that is the area where we get large number of patients now. So we need to now look at this. So I first published a paper, hot water epilepsy, a variant of reflex epilepsy in southern India. And this was published in one of the very highly rated epilepsy journal, Epilepsia. This was our first initial observation as a student. As a student, I started with this, and we looked at all our epilepsy cases in our institute and compared that with the hot water epilepsy. What we understood was 4.2% of the patients coming with epilepsy, complex partial seizure, and a generalized tonic-clonic seizure consist of this hot water epilepsy in our own setup. That means it is not uncommon. If you sit in our outpatient, any day I can show you at least 5 patients. 5 patients coming with hot water epilepsy can be seen any time. So that is the prevalence of this type of epilepsy. And what is this type of epilepsy? 2/3 of the patients come to us with complex partial epilepsy, which I will show you in a video. But 1/3 of the patient comes to you with a generalized tonic-clonic seizure, which all of you have seen on the roadside. A person falls down suddenly and gets a stiffness and also convulsive movement. And that is the type of epilepsy which occurs on a hot water bath in 1/3 of the patient. But what is interesting is about 10% of the patients are very, very interesting. And these patients, they pour water and water and water till they get the convulsion and fall down, because they enjoy that. They get a pleasure, they get a kick out of taking this hot water bath. As they get more and more water, they get a convulsion and fall down, because that is an intense pleasure they get with that. That is seen in 10% of the patient in our own series. So what do they come to us? They come to us with, when they take the hot water bath, suddenly the child stays a dazed look, irrelevant speech, and visual and auditory hallucinations, vertigo, the complex automatism, and a fear. That is the problem which these patients have. Here is a patient. I will be showing how it happens. As you can see here, he is a 12-year-old boy. And this is a common variety of giving a bath. And you see in the bathroom, we'll have a buckets with hot water and the cold water. You mix the water and then give it. The mother is giving the bath to this child. And this child is a 12-year-old. As he poured the water over the body first and then over the head, what is happening? He is confused. He doesn't know what he is talking. He is talking irrelevantly and he is not answering any questions. And look at his face now. Look at the face along with the mouth. He is doing certain movements which he doesn't know. And he is answering something irrelevantly. He is being asked who is that lady who is standing behind. He does not know. And he is totally confused, and he does with some movements with the hand. That is called automatism. And this is a typical complex partial epilepsy. And this boy gets every time when he takes the hot water bath. And you can see the eye, there is a nystagmus. There is an involuntary movement of the eyes, and the pupils were dilated during this time, thereby suggesting that it is a seizure phenomena. This type of seizure has been seen from all over the world. You have a series here, but ours was the largest series of 279 patients whom I have seen over the 4-year period and analyzed completely, and given the semiology part of it, what exactly happens in these patients. Most of them were children, but it is also seen in adults. Most of them get before 15 years of age. Youngest I had was a 2-year-old child. The 2-year-old child, oldest I had was 85-year-old man also. So we have extremes, but most of them occurs with a young age, and that is very, very important. Is it a benign one? The question which people ask. So we followed up. We followed up this series of cases, and you have series now from Mani, Dr. Subramaniam, Kurata, Miyayo, and myself. And what we saw was a very interesting one. 25% of these patients, when they continued to get this hot water epilepsy, afterwards they start getting epilepsy without the hot water bath. That is the problem. It is not that benign. So that means they start getting the epilepsy without even the hot water bath. And that is non-reflex seizure. So we started putting a hypothesis. What is the cause for this type of epilepsy? One of the hypothesis was a complex tactile and temperature dependent stimuli precipitated this epilepsy. Somebody said that there must be a lesion inside the brain in the temporal lobe, and that is producing epilepsy. That is the structural lesion. But we postulated a hypothesis, what is known as hyperthermic kindling. That means there is a kindling which is occurring in the brain due to the hyperthermia. Now we need to prove this point. It is very easy to put a hypothesis, but we need to prove. What did we do? We started going into an animal. So we started an animal experiment with a rat as the model. And the first animal model mimicking hot water epilepsy was developed, and that is the adult Wistar rat. And here the rats were given on the head the water with a syringing with the water, with the hot water, with a particular temperature. And then we looked at that the rat gets the seizure. The rat gets the seizure at a particular time after the temperature rises. So then we put the electrodes into the brain of these rats, into a particular region, which is the medial temporal lobe and a hippocampal area. And that electrode served both for recording the temperature as well as for the EEG. Then electro recording of the seizures were done by depth recording, and the seizure initiation, we noted that it is dependent upon a particular temperature. That is what we learned. We learned that when the rat temperature goes up to a particular area, then the rat gets the seizure. And that was a very, very classical one. And that is why we said that it is due to hyperthermia, and that rat produces a seizure. Here is a rat on the table where the stereotactically we place the electrodes into the brain, and on the rat we conducted the experiments and recorded the EEG. Because if you put the electrode on my head and give a bath, nothing can be recorded because the whole electrode goes away. But in the rat, we can put the electrode into the brain. I can't do it in the human being because it is an invasive procedure. So we did in the rat and then recorded that these are seizures. This is a seizure activity which is obtained from the brain, and that produces electrical seizure. And that is why we could do that. Then afterwards, what did we do? We sacrificed the rats and we looked at the hippocampus. And that can be done in the human being, it is impossible. But in the rat we could do. We said that there is a hyperthermia in the rat which produces hot water epilepsy, which is an animal model for this. And we then further worked on, and this is the brain of the rats. And the brain of the rat, which we could see here, this is a normal hippocampus, which you can see beautifully, all these parts of the hippocampus, which is a horse's brain. And then you can see here, after the hot water epilepsy, there is a definite damage which has occurred here, and whether this is a cause or the effect is the question, which is seen in the C2 area and C3 area of the hippocampus in the rat brain. So what could we see? Then we went on further, because our hypothesis was a hyperthermic kindling. The kindling is a very, very interesting phenomena. That means if you go on stimulating for these rats, we must be able to produce the rats without the hot water bath. Is it possible? Yes, that was possible. We started stimulating the rat every day. Nothing happened. Nothing happened. The rat continued to get a seizure. Beyond that, nothing. But what we did was, every alternate day we started stimulating. It is not every day, every alternate day. And then what we found was, by 14th day, the rat started getting a seizure even without the hot water bath in the cage, which was videographed and recorded. And the rat gets the seizure even before the earlier time. The latency comes down, the severity increases, the duration of the seizure increases, and then rats gets spontaneous seizure. That means there is a kindling. The brain was kindled to produce that. So I have an evidence now there is a kindling. But that is not sufficient. We have to prove that point. And that proving that point involves using a TIM staining. There is a stain which is called TIM stain, which was not available in India at that time. We have to import that and then look at the brain and then show whether there is a sprouting of the neuron. That means the nerve cells have to sprout. Then only we can say that the brain is kindled. So here is an evidence that there is a sprouting of the neurons which have occurred here. These are the neurons which have sprouted. This is a normal one in a contrast, and here you can see these neurons are all sprouted, thereby showing that there is a kindling in the rat. So we demonstrated hyperthermia, we demonstrated kindling in the rat. By understanding this is not sufficient. Now we went to the next one. Does it occur in the human being? That is the question all of you will be curious to know. So we postulated next hypothesis, and that hypothesis was, yes, there must be thermal regulation which is defective in human beings, and that is producing the hot water epilepsy. But does it occur in everybody? No. It occurs in a person who is susceptible to that, and that must be susceptibility to hyperthermia with an aberrant thermoregulatory system produces hot water epilepsy. This is our next hypothesis. So we went on to lay out an experiment. We started stimulating the people with a hot water bath. So all of us take the hot water bath. All my residents, my nurses were the guinea pigs for me. And they were all given that bathing has to be done. And we developed a thermistor. And a thermistor was putting into the pinna of the ear and record the temperature when they take hot water bath. And this was done experiment in our own bathing, in our wards now, and recorded the temperature. Nothing happens in the normal human being. When we take hot water bath, when we record the temperature, however hot it is, the temperature goes up by 0.5 degree, and then it comes back as soon as our bathing is over. Nothing happens. Then I have looked at the patients who are getting this hot water epilepsy, what they get as 1:1. That means every time when they take hot water bath, they get this seizure. They are easy to get that. Such patients we grouped and then started giving the bath in our own lab. And this thermistor was recorded, and it is connected to an oscilloscope and then recorded the temperature, what the temperature happens in these human beings. It was surprising. What we saw was very, very interesting. Here, these are the patients where the temperature shoots up. The temperature shoots up by 2.5 degree within a span of 2 minutes. Within a span of 1.5 to 2 minutes, the temperature of the person goes up and the person gets a seizure. That means it is almost like high temperature, like a fever when they take hot water bath. They don't complain, but we record that. When we record, what is important is the rapidity with which the temperature raises, produces a seizure. And at a particular temperature, these human beings start getting a seizure. And this was noticed very, very clearly. And repeatedly we did it, and then compared with a normal human being, and then it is all recorded, thereby showing that there is hyperthermia in these people which produces the seizure. That means what? Whether they have a febrile convulsions in the past. We looked at the history. 25% of our patients with hot water epilepsy had a history of febrile convulsion. And that was the history which we obtained. So we started now looking at this with rise in the temperature. Can we now do something about it? And this was the human experiments which we did. Aberrant thermoregulatory system in a susceptible individual which produces hyperthermic seizure. Possibly there is a kindling in the human beings also. The evidence is that they get non-reflex epilepsy as the time passes, which was not there. About 25% of them gets non-reflex epilepsy. So that means there is a kindling which is occurring, but nobody believes that there is kindling in the humans, so I cannot dare enough to say that there is kindling in the humans, but that is my own evidence here. But did we have any evidence for that? Yes, we did look at it. There are multiple factors which is responsible for hot water epilepsy. It is contact with the water, temperature of the water, the specific area of the stimulation, and repeated stimulation. These are the factors which are responsible for bringing the hot water epilepsy. We wrote a paper on trigger mechanisms in hot water epilepsy. At the same time, my colleague pathologist, Dr. Shankar, who was working in pathology department, looked at all his brains, and there were 4 brains of the people who had hot water epilepsy history. And then they have died for some reason. And when they died, their brains was with us. So we looked at 4 of these brains. As you can see here, these 4 people ages 15 to 53 years, and 2 are 12 years and 1.5 years. Duration was very long. They have type of seizures, and then they have died. 2 people because of the status epilepsy, and 2 others had incidental hypothalamic failure, one of the patient, post-operative, which I am sure Dr. Mathaya would have been very happy to know. Post-operative hypothalamic failures are known to occur, and vertebrobasilar insufficiency. So we had the brains, and we looked at the brain in these 4 areas, thereby showing that there is a damage which has occurred in these patients with hot water epilepsy. Then we looked at EEG of these patients in 70 people, out of which 55 were normal and 15, that is 21.4%, were abnormal. We had from the literature, and these are the cases with a classical spikes and foci across the anterior temporal region, which is seen in these patients with interictal EEG abnormality. There is a series which has been published, and now from 1965 onwards, but ours is one of the large series. There are abnormalities in the temporal spike, and in the Western literature, even they have put the caps and water immersion in the bathtub. They have recorded and shown that there are left anterior temporal focus, left hemispheric onset, and posterior frontal region. There are spikes in these, thereby suggesting that they are definite epilepsies which are arising from the hot water stimulation, and it is in the temporal region. Then somebody had proven point saying that there is an abnormal brain in this. So can we look at it? So we started doing an MRI. An MRI of these patients were done. Majority of them had normal, except that there were 2 people with a bilateral MTS, medial temporal sclerosis, and 1 had a left MTS. These were the patients who had a long time hot water epilepsy. That means whether it is a consequence rather than the cause for the epilepsy. That means repeated stimulation has produced the medial temporal sclerosis in these patients. But we still do not know what is the cause. That means I need to know if there is no anatomical abnormality, there has to be a functional abnormality. So how can I prove the functional abnormality? It is easy now, because there are what is known as SPECT scan. Ictal SPECT scan can be done. So we went ahead and searched for where is it available in Bangalore at that time, and in ictal SPECT scan, and we started doing. That means I can take these patients and put a needle and then give a bath, and as the bath is given, we can record where the blood flow increases, and that is an ictal periictal scan. So we used ethylene cysteine dimer as one of the radiopharmaceuticals which could be used, and then did a SPECT scan on these patients with hot water epilepsy. And what is interesting is here, we have a normal EEG on this patient with hot water epilepsy, normal MRI here, there are no abnormality, but we did a SPECT scan, and what we shown is a normal interictal scan interictal scans are here, it is normal, but you can see the difference between this and this. You can see the difference between this and this. You yourself can see that there is an abnormality with hyperperfusion, showed a left medial temporal hyperperfusion in this area when the hot water epilepsy occurred in these patients when the bath was given. Again, we repeated some more patient. This is another patient who has got a normal interictal EEG and an MRI, thereby suggesting that there is no structural abnormality. These are interictal scan and these are the ictal scan. And what we are seeing is definitely was a left lateral temporal hyperperfusion, thereby suggesting that there is a functional abnormality in these patients with hot water epilepsy occurring in the temporal area. And that is what we published here. The clinical and SPECT studies in the suggested temporal lobe as the seizure onset zone in some of the patients with hot water epilepsy. Then we thought that autonomic function should be abnormal in this, otherwise their thermal regulation will not be abnormal. So can we not do that? It can be done very easily. That means we need to demonstrate whether there is an abnormality in autonomic nervous system, which Mathai was very much interested in this area. So we started looking at that. We have the patients with epilepsy suddenly dying. That is known as sudden unexpected epileptic death syndrome, SUDEP, which we know that there are patients who have epilepsy, suddenly one fine day they found dead. And that occurs even in children, which is called cradle deaths or SUDEP, SUDEP. So what is the cause for SUDEP? One of the major causes they found is because of the autonomic imbalance in these patients with epilepsy. Do we have a proof on that? There are many variety of autonomic manifestations which is seen in epilepsy. So this is known, the gustatory, which could be cardiac abnormalities. These are all very well known in the patients with epilepsy. So we started looking at normal first, a cardiac autonomic function in a drug-naive, newly diagnosed epilepsy patient. We published this paper that there is definite interictal subtle cardiac autonomic dysfunction which occurs in patients with epilepsy, which is a very well known phenomena, which we did it in patients with seizures. Then these are the maneuvers which we did: Valsalva maneuver, isometric hand grip test, then tilt table test, which Mathai was shown in his laboratory. And we have an autonomic laboratory in our own setup, and we did that, and we looked at the hot water reflex epilepsy, and thereby we showed that there is a long work. This is our objective was this, and we did this. Pure hot water epilepsy patients, we took 45 patients, hot water epilepsy with a spontaneous complex partial seizure, that means non-reflex seizure, 25 patients, unprovoked spontaneous complex partial seizure, 40 patients, and a healthy volunteers, 50 patients and 50 subjects. And then we looked at them and conducted autonomic function tests, heart rate variability, blood pressure variability, baroreflex sensitivity tests. All these things we did in this group of patients, and then this is the 45 patients who had pure hot water epilepsy, and then we did that compared with a healthy volunteer, and various autonomic function tests were done. And all these parameters have been mentioned here, and statistical significance have been mentioned compared with the 45 controls. And thereby we showed that there is a heart rate variability in these patients, and there is a variation with the blood pressure, and there is a variation with baroreflex sensitivity, and thereby suggesting that in this cardiac autonomic function test, there is abnormality in patients with hot water epilepsy with an altered sympathetic vagal balance. That means there is an autonomic imbalance in these patients with hot water epilepsy, and that is resulting in the temperature regulation problem, and that temperature regulation problem leads to hyperthermia, and hyperthermia leads to seizure. But one other question we had, that question was, we were mentioning that there is a susceptibility. So we need to now, can we take this understanding to a patient and treat them? It is a very, very next question which we ask. So how do we treat? Can we now say, people were asked to say not to take hot water bath. Can you believe? If you can say that not to take hot water bath, it is like a person who is taking alcohol. Very easy answer, stop drinking alcohol. I can preach 100 people, how many will accept it? They won't accept it because they get pleasure. Similarly, these patients, I said that they get pleasure out of hot water bath. That is a very, very difficult. Then the people were asked to say that don't bathe yourself, take a wet towel and rub the head. That is what that was the treatment which was given earlier. Then we were wondering, when I know there is a hyperthermia, can I use paracetamol or aspirin tablet? It doesn't work. You know why it doesn't work? Because they are normal temperature in the beginning. The temperature goes up within 2 minutes time and they get the seizure. So that doesn't work. But we have a model of febrile convulsion, where in the febrile convulsion, people are given benzodiazepines and they get prevented. Taking that cue, we started working on using benzodiazepines, and now we have all established, various experiments have been done. We now ask these patients to take one clobazam tablet, that is 5 milligram for children and 10 milligram for adult, 1.5 hours before taking hot water bath on the head on that day. And if they take it, they don't get any seizure and they enjoy the bath. And they have come back testing me, stopping the drug, and they get a seizure. And that is my proof here. An intermittent clobazam prophylaxis prior to head water bath might be a preferred mode of treatment, and only anti-epileptic drugs are required if they have associated non-reflex epilepsy. Otherwise, they don't require anti-epileptic drugs. And this is what now we practice, and this is a method of treatment which has been published, and this is the results there, and patients are very happy. They just take the tablet, because most of them take the hot water bath once in a week, sometimes once in 3 days or once in 10 days. Those are the people, when they take hot water bath over the head, they take this tablet and they are enjoying their bath without any problem. And this was an understanding because of our animal model and developing. I need to prove another point, whether there is a susceptibility. Susceptibility individual means genetics. So that means, is there a genetics of hot water epilepsy? Do I have about 5 minutes more? Yeah. So I looked at the 304 patients with the phenotypic characteristics of hot water epilepsy, complex partial seizure with secondary generalization, and primary seizure. So we looked at it, these 304 people, and trios, that means the patient and the father and the mother, or the family history when it is positive. There were 67 people with the family history positive. Out of the 67, we had small family, that is 2 members affected, or 41. Medium family, there were 3 members affected, was 12. But interestingly, I had large family with 4+ people affected. But I was looking for these people where there is a strong family history of hot water epilepsy. So we started jumping at that, and this is the family where I looked at. I went to the village and collected all the family members' blood and also the pedigree chart. We have here 14 members affected here, and all the family members, starting from a 2-year-old child to a 94-year-old man who had the hot water epilepsy. We collected the blood sample, both affected and unaffected, and started looking at it. First thing what we wanted to look at is there a febrile convulsion gene. Whether, because this is like febrile convulsion, can I rule out a febrile convulsion genetics? We know the genetics of febrile convulsion, so we have the genes, that is called Feb1. Feb1 gene was looked for, it is not there, it was negative. Then we looked at Feb2, another gene, that is negative. Then we did the third one, febrile convulsion gene, that is also negative. That means all febrile convulsion genes were not there in our hot water epilepsy. Then what should we do? There is no other way but to go for whole genome scan analysis. That means whole genetic analysis has to be done in this family, then only we can do that. So we started looking at the whole genome scan, and in this family we have an association with 10th chromosome, 10q21 to q23. And this is the area which is the genetic abnormality which we saw in this chromosome, in this family. Then another family we started, haplotype analysis was done, and then we have a smaller number with 2 members and 3 members affected. We added their blood and looked at the larger families, and then we looked at that there is an additional locus. This was the locus at 10q21, q23 was the first association in those families, both with the large family and the small family. But we were not happy that this could be an association. Is this the single gene disorder? No. We started looking at, these are the genes which are available. There are potassium, large conductance, calcium activated channel, voltage dependent anion channel, solute carriers, and glutamate receptor channels are available in this area. So we started looking at it, and then we published that paper, and we next looked at another family, and this is a larger family with 7 members affected with hot water epilepsy. And in this family, we added few more families with the smaller families with 3 member, 4 member affected, and looked at the same 10th chromosome. The 10th chromosome was not available, and here there was another chromosome which is at the chromosome 4. And in the chromosome 4, at this area, so various genes were there, and this is the area which was abnormal, and my pathologist, my genetist looked at this gene in carefully, and what we see here, there is a gene for partial epilepsy, there is a gene for familial medial temporal lobe epilepsy, and we have a gene in this area which is a hot water epilepsy gene. That means it is an epilepsy gene in that chromosome which is responsible for the hot water epilepsy. But we started looking at, can we have what is that gene? Is it possible to get the gene out? This is an association now. We started looking at the various families. This has been published now, and this is a family which is a larger family with a smaller families added together, and we looked at this gene, and there is another chromosome which is the 9p 24.3 to 23. This is the area which was abnormal, and this is a hot water epilepsy area in that particular chromosome. We have a epilepsy familial temporal lobe, epilepsy idiopathic generalized susceptibility in this part of the gene, and epileptic encephalopathy early infantile in this part of the gene. So this is the whole chromosome, which is a large one, and this area is responsible for hot water epilepsy. This region harbors about 60 genes, out of which 24 are expressed in the brain. So we started looking at it, and then we were lucky, and this is the gene which we could identify now. And this is the first time in India we are able to isolate the gene, and this new variant was not found in 540 control chromosomes from the normal individuals, and the gene is a transporter of glutamate at the pre and the postsynaptic neuronal terminal, and this is the gene, and this is the gene which we could identify, and it is a missense variation, and we have published in this family, and these genes are called SLCA1 or EAAT3 gene, and there are 3 types of genes which are available, and this is present in the neuronal somata dendrites but not enriched in the spine, and this is the gene which is responsible for hot water epilepsy. That means we have now a gene which has been responsible for hot water epilepsy, and this is a glutamate transporter receptor gene abnormality. So in summary, hot water epilepsy is frequently reported from South India, though had a worldwide occurrence. An animal model mimicking hot water epilepsy has been developed and demonstrated that these are hyperthermic seizure with kindling. It is a type of hyperthermic seizure in humans with a possible aberrant thermoregulation in genetically susceptible individual. Functional imaging has been done, demonstrated ictal abnormality in the medial temporal structure. Cardiac autonomic function tests demonstrated abnormality in sympathetic vagal tone, and then they can be managed using a prophylactic benzodiazepine just before having a hot water bath akin to febrile convulsions. Only those with a non-reflex seizure require AED therapy. I started my lecture saying that some people doubted whether such an entity exists or this is a functional phenomena. I proved the point, and at the end in 2004, the classification of hot water epilepsy has been included in the International Classification of Epilepsy now, and this is ILAE proposed classification which is included, and now this is there as a stamp of that is in hot water epilepsy is a reflex epilepsy. And in the genetics, 12 to 27% of hot water epilepsy patient has a positive family history, and hyperthermic seizure first ruled out association with febrile convulsion genes. Whole genome scanning of the hot water epilepsy patient in large family yielded multiple association with 10q, 4q and 9p chromosomes, thereby confirming that hot water epilepsy is a polygenic complex epileptic disorder. And exome sequencing yielded a gene which is known as SLC1A1, a gene, a glutamate transporter gene which is responsible for this. So we still propose to do some more work, that is a receptor physiology, channelopathies, and I'm now got a PET MRI scan in our institute for the first time in the country, and we plan to use this and to look at PET MRI in these patients, seeing the abnormality, because we can directly see the radiopharmaceutical tagging on and then look which region is abnormal, and genetic epidemiology to look at which area, why they are more common in certain areas of the country. And this I owe it to my teacher, Professor K.S. Mani, who described the hot water epilepsy much earlier, and I only picked it up from him and started working on to find the answers, because it was considered as a doubtful entity. And here all the people, Dr. Gautam, who is a physiologist, was in MS Ramaiah College, who worked with me, Shankar, who is a pathologist, Anuranjan Anand, who is a genetist, Sanjeev Sinha, who is my colleague, who is working with me. This is Satyaprabha, who is a physiologist, who is working on autonomic functions, and these are my PhD and doctoral students who have worked with me to understand about the hot water epilepsy. And that is, I leave you with my flora and fauna of NIMHANS campus, and thank you for assistance.